All right, let's dive into industrial biotechnology. Getting ready for that semester 6 exam, huh? I know, it can feel like a lot. That's so many processes to remember.
It's definitely a challenging subject.
But that's why we're here. This question bank deep dive is going to help you nail those concepts.
Absolutely. And I'm here to help break it all down.
Perfect. So with our biotech brain joining us, let's tackle those exam questions.
That's good.
Okay, first up, a question you're almost guaranteed to see. Explain the production of citric acid.
Citric acid, it's in practically everything from sodas to candy, right?
Right, it's everywhere.
Yeah.
But have you ever thought about how it's actually made?
I'll admit, I haven't given it much thought, but I'm ready to learn.
Well, get ready for the fascinating world of Aspergillus niger. This fungus is a rock star in industrial biotech.
Aspergillus niger, huh?
Yeah, it's the key player in citric acid production. And the process it uses is called submerged fermentation.
Submerged fermentation. Basically, imagine giant tanks filled with a nutrient broth where Aspergillus niger is basically having a feast producing citric acid as a byproduct.
It's like a giant fungal buffet.
Oh, okay.
So we've got these Aspergillus niger cells swimming around in this broth, churning out citric acid.
Right. But here's the thing. To really get this process going, to maximize citric acid production, we need to control the environment precisely.
So it's not just about providing those fungi with food. We need to create the perfect environment for them to thrive.
Exactly. We're talking about pH, temperature, oxygen levels. They all play a crucial role. And understanding why is key for your exam.
Okay, let's break it down. Let's start with pH. Why is that so important?
Well, Aspergillus niger likes things a bit acidic.
Acidic.
Yeah, the optimal pH range is between 2.5 and 3.5, and there's a reason for that. This low pH actually helps prevent contamination.
Contamination.
Yeah, from other microorganisms that might want to crash the party and compete for resources.
Ah, so it's like a defense mechanism for Aspergillus niger, keeping those unwanted guests out of the fermentation pool party.
Precisely. That low pH gives Aspergillus niger a competitive edge.
So low pH helps keep things clean and efficient. Okay, what about temperature? What happens if things get too hot or too cold for our fungal friends?
Well, Aspergillus niger is a bit of a Goldilocks. It needs the temperature just right.
Not too hot, not too cold.
Exactly. The optimal range is 28-30 degrees Celsius.
Celsius, okay.
At this temperature, its metabolism is humming along, producing citric acid like a champ. But if it gets too hot, its enzymes, those crucial proteins that do all the work, can start to denature.
Denature. What does that mean?
It means they lose their shape and can't function properly. It's like if you boiled an egg, the proteins change and solidify.
Ah, got it. So too hot and the enzymes basically break down.
Right, and too cold, well, its metabolism slows down and citric acid production drops. It's like a car engine that's not warmed up properly. It won't run efficiently.
So maintaining that optimal temperature range keeps those fungal enzymes happy and those citric acid factories churning. Okay, last but not least, let's talk about oxygen. All living things need to breathe, right?
Absolutely, even fungi.
So how do we ensure those Aspergillus niger cells are getting enough oxygen in those massive fermentation tanks?
Well, Aspergillus niger is an obligate aerobe,
Obligate aerobe.
meaning it needs oxygen to survive. It uses oxygen for respiration, breaking down sugars to release energy.
Right, respirate. That's how cells generate energy.
Exactly. And that energy is needed for all cellular processes, including citric acid production.
So no oxygen, no citric acid.
Pretty much. That's why in industrial fermentation, the broth is constantly supplied with oxygen, ensuring those Aspergillus niger cells have enough oxygen to power those citric acid factories.
It's like giving those fungi a constant oxygen fuel supply.
Precisely.
This is fascinating. But wait, there's more. We need to talk about nutrient limitation, which seems counterintuitive. You're saying we restrict nutrients to increase citric acid production. How does that work?
It might seem strange, but yes. By restricting certain nutrients, we can actually trigger a metabolic shift that favors citric acid production.
A metabolic shift.
Yeah, think of it like this. You have a metabolic pathway, a series of reactions that leads to the production of citric acid.
Okay, so a step-by-step process within the cell.
Okay. And one of the key players in this pathway is an enzyme called aconitase.
Aconitase. Okay, what does it do?
Aconitase typically consumes citrate, a precursor to citric acid, in a process called the Krebs cycle. It's a central metabolic hub in the cell.
So aconitase uses citrate for energy production.
Essentially, yes. But by limiting certain nutrients, like phosphate and some trace metals, we're reducing aconitase activity.
We're putting the brakes on aconitase.
Exactly. And that creates a bottleneck in the pathway.
A bottleneck.
Yeah, citrate can't be consumed as quickly, leading to a buildup, and that excess citrate is then channeled towards citric acid production.
So it's like redirecting traffic on a metabolic highway, forcing those citrate molecules to take the citric acid exit. Brilliant.
Precisely. It's all about manipulating those metabolic pathways to our advantage.
Incredible. So we've got our Aspergillus niger happily producing citric acid in this carefully controlled environment, but then we need to actually extract it from all that broth, right?
That's right, and that brings us to downstream processing, a whole other adventure in itself.
Okay, let's save that adventure for another time. But before we move on, I want to go back to that nutrient limitation strategy. You mentioned phosphate and specific trace metals.
Yeah.
Why those specific nutrients? What makes them so crucial for manipulating citric acid production?
Good question. It comes down to their roles in cellular metabolism. Phosphate, for instance, is a key component of ATP.
ATP, the cellular energy currency.
Exactly. And it's also involved in regulating enzyme activity. So by limiting phosphate, we're kind of creating a slight energy shortage, encouraging those fungal cells to focus on citric acid production.
So it's like we're giving those cells a little nudge, saying, hey, focus your energy on making citric acid.
Precisely. And those trace metals, like iron and manganese, are important for certain enzymes, including that aconitase we talked about.
Right, aconitase, the citrate consumer.
Exactly. By limiting those trace metals, we're further reducing aconitase activity, boosting that bottleneck effect, and increasing citric acid production.
It's amazing how much we can control those microbial factories by adjusting their diet. Now let's shift gears and tackle another question that's likely to be on your exam. Describe the extraction and purification of citric acid.
Sounds good.
This seems like a multi-step process. Where do we even begin?
Well, the first step is to separate the fungal biomass, the mycelium, from the fermentation broth.
Okay, so we need to get all those Aspergillus niger cells out of this citric acid solution.
Right. Think of it like separating the swimmers from the pool water.
Got it. But how do we actually do that on an industrial scale?
Filtration is a common technique. Large rotary vacuum filters or pressure filters are used. They're basically giant sieves that separate the solids from the liquid.
Okay, so we're filtering out those Aspergillus niger cells. What happens next?
Well, to make the filtration process even more efficient, we often use filter.
Filter ACE.
Yeah, things like diatomaceous earth. It's a natural material that helps prevent clogging and keeps the process running smoothly.
So it's like adding a helper to the filter to keep things flowing.
Exactly. And once we have that clarified citric acid solution, there are two main approaches to extract the citric acid itself.
Two approaches.
Yeah. The traditional method, which is still widely used, involves adding calcium hydroxide, also known as lime, to the solution.
Okay, what does that do?
Well, this reacts with the citric acid to form calcium citrate. And calcium citrate is insoluble, so it precipitates out of solution as a solid.
So it's like we're pulling the citric acid out of solution by turning it into a solid.
Precisely. We're essentially converting it into a form that's easier to separate.
Clever. But then how do we get from those calcium citrate crystals back to pure citric acid?
That's where sulfuric acid comes in.
Sulfuric acid.
Yep. We add it to the solution, and it reacts with the calcium citrate, converting it back to citric acid.
So it's like a chemical switcheroo.
Exactly. We swap calcium for hydrogen ions to get our citric acid back, and the byproduct is calcium sulfate, also known as gypsum.
Okay, so that's the traditional method. You mentioned a modern alternative, right?
Yeah, and that involves using organic solvents.
Organic solvents.
Yeah. This method takes advantage of the fact that citric acid is more soluble in certain organic solvents than it is in water.
So we're basically using a solvent that's better at grabbing onto those citric acid molecules.
Exactly. We mix the citric acid solution with the solvent, and the citric acid preferentially moves into the solvent layer. Then we can separate the two layers, leaving us with a concentrated citric acid solution in the solvent.
It's like citric acid playing a game of choose your solvent, and it prefers those organic ones. But why is this method gaining traction? What's the advantage over the traditional precipitation method?
Well, one major advantage is environmental friendliness. The traditional method generates a lot of gypsum byproduct.
Right, from that calcium sulfate.
Exactly. And that can be a disposal challenge. Solvent extraction, on the other hand, allows for solvent recovery and recycling.
Ah, So it's a greener approach to citric acid extraction.
Exactly. And once we've extracted the citric acid, whether through precipitation or solvent extraction, we move on to purification.
Purification to get that super pure citric acid used in food and everything else, right?
Right. We need to remove any remaining impurities, things like pigments, proteins, and other unwanted molecules.
So it's like a polishing process.
Exactly. And there are several steps involved. Decolorization is often the first step, using activated carbon.
Activated carbon.
Yeah, it's like a super sponge, absorbing those color impurities.
So it's like giving that citric acid solution a charcoal detox.
Exactly. Then we often use ion exchange chromatography. This technique separates molecules based on their charge.
So it's back to separating molecules based on their properties.
Right. We use tiny beads with specific charges, and molecules with opposite charges stick to the beads while others pass through. This helps us separate the citric acid from any remaining charged impurities.
It's like a molecular sorting hat.
Mm.
And finally, the grand finale, crystallization.
Yes, crystallization. This is where we get those beautiful, pure citric acid crystals. We concentrate the purified solution, and as it cools, those citric acid molecules start to form those highly ordered crystalline structures.
It's like a molecular dance party, where those citric acid molecules are getting cozy and forming those sparkly crystals. This whole process from submerged fermentation to extraction to purification is truly fascinating. But I have one final question about citric acid production. You mentioned Aspergillus niger is a workhorse in industrial biotech. Are there other microbes that can produce citric acid?
There are, but Aspergillus niger has some key advantages. It's a very efficient producer, meaning it can churn out large quantities of citric acid. It's also relatively easy to cultivate.
Easy to cultivate.
Yeah, it's not too picky about its growth conditions. And importantly, it's considered safe for food and industrial applications.
So it's like a trifecta of efficiency, ease of use, and safety. No wonder Aspergillus niger is the champion of citric acid production.
Exactly. And remember, for your exam, focusing on those key characteristics of Aspergillus niger that make it such a good citric acid producer will really impress your professors.
Excellent point. Okay, ready to move on to another essential biomolecule.
Absolutely, bring it on.
Perfect. Let's tackle this question. Explain the production and extraction of vitamin B12 and B2. I know these B vitamins are essential for life, but I'll admit I'm a bit clueless about how they're actually made.
Well, get ready for another fascinating journey into the world of microbial factories. Both B12, also known as cobalamin, and B2, also known as riboflavin, are produced industrially using specific microorganisms.
So it's like each vitamin has its own preferred microbial production team. Who are the key players in the B12 and B2 leagues?
For B12, we've got a few bacterial stars. Pseudomonas denitrificans and Propionibacterium species are commonly used. Pseudomonas denitrificans is known for its rapid growth and high B12 yields.
So it's like the speed demon of B12 production.
Exactly. But Propionibacterium species, particularly Propionibacterium freudenreichi and Propionibacterium shermanii, are gaining traction, especially in the food industry.
Why the preference for Propionibacterium in food applications? What makes them stand out?
They have a secret weapon,
Yep.
GRAS status. It stands for Generally Recognized as Safe.
GRAS status, so it's like a safety stamp of approval from regulatory agencies.
Exactly. It means they're considered safe for use in food, crucial for anything that's going to end up on our plates.
Makes sense.
Well-
Okay, what about B2 production? Who are the star players in that microbial league?
For B2, we've got a fungal champion, Ashbia gossipii. This filamentous fungus is a riboflavin-producing powerhouse.
So we've got Pseudomonas denitrificans, or Propionibacterium species for B12, and Ashbia gossipii for B2. Now, how do these microbes actually produce those essential B vitamins?
Well, fermentation is the key, just like with citric acid. But the specifics vary depending on the microorganism and the vitamin.
Okay, so different microbes, different fermentation strategies.
Right. For B12 production, for example, the oxygen situation can be a bit tricky. Historically, B12 production relied on anaerobic fermentation, meaning those microbes were working in the absence of oxygen.
No oxygen, but we just talked about how important oxygen is for respiration and energy production.
Yes, but some microbes have evolved to thrive in oxygen-free environments. They use alternative pathways to generate energy.
Interesting. So they've adapted to a different energy source.
Exactly. And many modern processes, particularly those using Pseudomonas denitrificans, are actually aerobic or microaerophilic.
Aerobic means they need oxygen, right? And what about microaerophilic?
Microaerophilic means they need only a small amount of oxygen. Some microbes can even switch between aerobic and anaerobic respiration depending on the conditions.
Wow, so they're like microbial chameleons, adapting to different oxygen environments. What about those strictly anaerobic producers, like Propionibacterium species? How do they manage B12 production?
They often use a two-stage fermentation process. First, an aerobic stage for rapid biomass growth. It's like giving them a feast to build up their cell numbers. Then, a shift to anaerobic conditions to encourage cobalamin synthesis.
It's like a carefully choreographed microbial dance, changing the oxygen levels to cue different stages of the production. Okay, what about B2 production using Ashbiogossypii? Is oxygen a factor there?
Ashbrigossyp is an obligate aerobe, just like Aspergillus niger, so it needs a constant oxygen supply throughout the fermentation process.
Okay, so we need to keep those Ashbiogossypii cells well oxygenated to keep those riboflavin factories running. Now let's talk about the fermentation media, the nutrient broth that feeds those B vitamin producing microbes. What are some of the key components in these vitamin boosting broths?
For both B12 and B2 production, a good carbon source is essential. This provides the primary energy source for the microbes. Glucose is a common choice.
Glucose is simple sugar.
Exactly. Corn steep liquor, a byproduct of corn wet milling, is another popular choice. It's rich in various nutrients, including sugars, amino acids, and vitamins.
So it's like a nutrient-packed supplement for those microbes.
Right. And molasses, a byproduct of sugar refining, is another option. It's packed with sugars and other valuable nutrients.
So we're essentially providing a balanced diet for those microbes, ensuring they have the energy and building blocks they need to grow and produce those B vitamins.
Precisely. And besides the carbon source, we need a nitrogen source.
Nitrogen.
Yeah, to provide the building blocks for proteins and other nitrogen-containing molecules. Ammonium salts, yeast extract, and again, that versatile corn steep liquor are often used.
It's all about providing those essential amino acids for those busy microbial factories.
Exactly. And we can't forget about the essential minerals and trace elements. These micronutrients play crucial roles in various cellular processes, often acting as cofactors for enzymes involved in B vitamin biosynthesis.
So it's like adding a pinch of mineral seasoning to that microbial recipe. But why are these minerals so important? What do they actually do?
They help those enzymes function properly. For B12 production, for example, cobalt is particularly important.
Cobalt.
Yeah, it's an essential component of the cobalamin molecule itself. So we need to add cobalt salts to the media to ensure those microbes have enough cobalt to build those B12 molecules.
It's like we're giving those microbes the right tools to build those B12 molecules.
Exactly. And for B2 production using Ashbiogossypii, biotin is often added to the media.
Biotin.
It's a vitamin itself, and Ashbiogossypii needs an external source of it to thrive.
So it's like giving those Ashbiogossypii cells a little extra vitamin boost.
Precisely. All these ingredients work together to create the perfect environment for those B vitamin producing microbes to thrive.
It's amazing how much goes into designing those microbial factories. It's like a delicate balancing act, making sure all those ingredients are present in just the right amounts. But after all that fermentation magic, how do we actually extract those precious B vitamins from the broth?
Well, understanding the location of those vitamins within the microbial cells is key. B12, for instance, is primarily intracellular.
Intracellular meaning it's trapped inside those microbial cells.
Exactly. So we need to break open those cells to release the B12.
Ah, so it's like a microbial jailbreak. How do we do that?
There are several methods for cell disruption, each with its own pros and cons. Mechanical methods, like high-pressure homogenization or bead milling, physically crush those cells. Chemical methods use solvents or detergents to disrupt the cell membranes. And enzymatic methods use specific enzymes, like lysozyme, to degrade the cell walls.
It's like we got a whole arsenal of tools to break open those cells and release those B12 molecules. What about B2? Is it also trapped inside those cells?
B2 is a bit more versatile. It can be both intracellular and extracellular. This means it can be found both inside and outside the cells.
So we might need to use those cell disruption techniques for B2 as well. But if a significant amount is already floating around in the broth, we can skip that step.
Exactly. And whether we're extracting D12 or B2, purification is essential to get that high purity vitamin product.
Purification to remove any remaining impurities, right?
Right. And both B12 and B2 purification often involves a combination of techniques: solvent extraction, chromatography, and crystallization. We've talked about those before.
Right. Those seem to be the all-star purification techniques in industrial biotechnology.
They are. And each technique is chosen based on the specific properties of the B vitamin and the desired purity level.
It's like choosing the right tool for the job. Each purification method has its own strengths and weaknesses.
Exactly. And now that we've covered the basics of B vitamin production, let's move on to our next challenge.
Sounds good. What's next on our microbial adventure list?
Let's explore the world of enzymes. Specifically, let's tackle this question. Explain the upstream process of enzyme production.
Upstream process. It sounds like we're heading upstream on a river of knowledge.
We are. And just like any journey, the first step is crucial: microorganism selection and strain development. We need to choose the right microbial workforce for our enzyme production factory.
Okay, so we're looking for a microbial all-star, a top performer in the enzyme production game.
Exactly. And several factors come into play when choosing the best microbe for the job. First, we consider the source of the enzyme. Is it naturally produced by bacteria, fungi, or yeasts?
So we need to know where this enzyme naturally comes from.
Exactly. Each type of microorganism has its own advantages and disadvantages. Bacteria like Bacillus subtilis and Escherichia coli are often used because they grow quickly and we understand their genetics well.
So they're like the fast and reliable workers in the microbial world.
Exactly. For some enzymes, though, fungi might be preferred, like Aspergillus niger or Trichoderma ricei, especially if the enzyme is naturally secreted into their surroundings.
Secreted, meaning released from the cell.
Exactly. This simplifies downstream processing because we don't have to break open the cells to extract the enzyme.
That makes sense. So it's like choosing a microbe that readily shares its enzymatic treasures.
Precisely. And for some eukaryotic proteins, yeasts like Saccharomyces cerevisiae are preferred. They offer advantages in terms of protein folding and modifications, which can be crucial for proper enzyme function.
So it's like choosing the right tool for the job: bacteria, fungi, or yeast, each with its own strengths and specialties. But even after we've chosen our microbial all-star, there's still room for improvement, right? That's where strain improvement comes in.
Absolutely. Naturally occurring microorganisms might not produce enzymes at the levels we need for industrial applications, or they might have other undesirable characteristics.
Undesirable characteristics.
Yeah, things like being sensitive to certain conditions or producing unwanted byproducts. And that's where strain improvement comes in.
It's like putting those microbial athletes through a rigorous training program to enhance their performance.
Precisely. We want to boost those enzyme yields, improve stability, and eliminate those pesky byproducts.
So how do we train those microbial athletes?
Well, there are two main approaches: classical and modern.
Okay, what's the difference?
Classical strain improvement techniques, like random mutagenesis, have been used for decades. This involves exposing the microorganism to mutagens.
Mutagens.
Yeah, things that cause mutations in DNA, like UV radiation or certain chemicals.
Okay, so we're basically inducing mutations, hoping for one that improves enzyme production.
Exactly. We then screen for those rare mutants with enhanced characteristics.
So it's like playing microbial roulette, hoping to hit the jackpot with a superproducer. What about the modern approach?
Modern biotechnology uses recombinant DNA technology and genetic engineering.
Ah, so we're getting more precise with our modifications.
Exactly. We can clone the gene for the target enzyme and optimize its sequence. We can add signal sequences for secretion and even modify metabolic pathways to increase precursor supply.
It's like we're rewiring those microbial factories to maximize enzyme output.
Precisely. These modern techniques offer greater precision and control.
Impressive. But even with the most optimized microbial strain, we still need to provide the right environment for those cells to thrive and produce those valuable enzymes. And that brings us to media design and optimization, right?
Right. It's all about creating the perfect recipe for those microbial chefs.
Okay, let's dive into that microbial recipe book. What are the essential ingredients?
The core components of any fermentation media are the carbon source, the nitrogen source, and mineral salts. The carbon source provides the energy and building blocks for cell growth and enzyme production. Cost-effective sources like starch, sugars, and agricultural byproducts are often used.
So it's like giving those microbial engines the fuel they need to power enzyme production. What about the nitrogen source?
The nitrogen source provides the building blocks for proteins and enzymes. We often use ammonium salts, yeast extract, peptone, or those versatile agricultural byproducts.
It's like a protein-packed feast for those microbial workers.
Exactly. And we can't forget those essential minerals and trace elements like phosphates, magnesium, iron, zinc, and many others.
What's the role of those minerals?
They act as cofactors for enzymes, ensuring those catalytic reactions run smoothly, and they also contribute to a healthy cellular environment.
So it's like making sure those microbial workers have all the tools and equipment they need to perform their enzymatic tasks.
Precisely. And for some enzymes, we might need to add specific vitamins or growth factors to the media, providing those essential nutrients that some microorganisms can't synthesize themselves.
That's like giving those microbes a little extra nutritional support. But with so many ingredients and variables, how do we determine the optimal media composition?
That's where media optimization comes in.
Media optimization.
We can use classical methods like systematically varying one component at a time, or more sophisticated statistical methods to simultaneously optimize multiple components.
So it's like a scientific process of experimentation and fine-tuning.
Exactly. We're looking for that perfect balance of ingredients to maximize enzyme production.
But before we move on to the main fermentation stage, there's one more crucial step we need to cover: inoculum preparation.
Right. Inoculum preparation is all about setting the stage for a successful fermentation.
So it's like getting those microbial athletes ready for the big game.
Exactly. We need to make sure we have a healthy, active, and contaminant-free population of microorganisms to start the process.
Okay, walk us through that preparation process. What are the key steps involved in getting those microbial athletes ready for the main event?
It typically involves a series of scale-up steps, starting with a pure culture and gradually increasing the volume through a series of transfers.
Okay, so we're gradually increasing the population size.
Right. We start with a small, pure culture in a test tube or flask, and then transfer it to a larger flask, and so on, until we have enough to inoculate the production bioreactor.
So it's like taking those microbial seedlings from a small pot to a larger one before finally planting them in the field.
Exactly. And throughout this process, we need to maintain purity, making sure there's no contamination.
Contamination could ruin the whole batch, right?
Exactly. We use various techniques to check for unwanted microbes, like microscopic observation and streaking on agar plates.
So it's like a microbial security checkpoint, ensuring only the authorized microbes get into the fermentation facility. But once we have a sufficient volume of that pure culture, what happens next?
Well, depending on the scale, we might transfer the culture directly to the production bioreactor. But for large-scale fermentations, we often use a seed fermenter.
A seed fermenter.
It's a smaller bioreactor that mimics the conditions of the production bioreactor. It helps generate a larger volume of inoculum and gets those cells acclimated to the conditions they'll face in the main bioreactor.
Ah, so it's like a pre-game warm-up for those microbial athletes, getting them ready for the main event.
Exactly. And this careful preparation, from selecting the right microbe to scaling up the culture and optimizing the media, sets the stage for a successful fermentation and high enzyme yields.
This is all incredibly fascinating. But I'm ready to move on to a specific enzyme example. Let's talk about penicillin. I'm sure our listeners are eager to learn about the production of this wonder drug.
Let's do it. Penicillin is a classic example of industrial biotechnology in action, and its production process highlights many of the key principles we've been discussing.
Perfect. Let's tackle this question. Describe the fermentation of penicillin. Now, I know penicillin is produced by a fungus, specifically Penicillium chrysogenum, but what are the specifics of the fermentation process?
Well, penicillin production is a testament to the power of strain improvement and optimized fermentation techniques. It all starts with Penicillium chrysogenum, a filamentous fungus that revolutionized medicine.
Penicillium chrysogenum, the penicillin pioneer.
Exactly. But we're not just using any old Penicillium chrysogenum strain. Decades of research have led to highly productive strains, specifically for industrial production.
So it's like scientists have bred a super productive penicillin-producing champion. How did they achieve that? What were some of the milestones in this strain improvement journey?
It all started with those classical random mutagenesis techniques we discussed earlier. Scientists used UV radiation, X-rays, or chemical mutagens to induce mutations in the original Penicillium strain.
So they were basically scrambling the genetic code, hoping for a lucky mutation that would boost penicillin production.
Exactly. They would then screen those mutated strains to find those rare gems that produced more penicillin. One particularly famous strain, Penicillium chrysogenum NRRL 1951, actually came from a moldy cantaloupe.
A moldy cantaloupe. That's a pretty humble origin story for such a revolutionary drug.
It is. And that discovery marked a significant step forward in penicillin production. But the journey didn't stop there. Further rounds of mutagenesis and selection led to even higher yielding strains. These improved strains weren't just better at producing penicillin, they were also easier to handle in bioreactors.
Easier to handle. What does that mean?
Well, some strands grew faster, some had less filamentous growth, which can make the broth too thick and difficult to mix, and some had better morphology.
Morphology.
Yeah, the shape of those fungal cells. And all of these improvements led to more efficient penicillin production.
So it's not just about maximizing penicillin output, it's also about optimizing those fungal factories for industrial scale production.
Exactly. And in the modern era, recombinant DNA technology and genetic engineering have taken center stage, allowing for targeted improvements.
So we're not just blindly inducing mutations anymore. We're specifically engineering those penicillin-producing pathways. What are some examples of these genetic modifications?
Well, one approach is pathway engineering. This involves modifying the penicillin biosynthetic pathway to increase flux towards penicillin production. For example, we can overexpress key enzymes in the pathway, like isopenicillin N synthase, which catalyzes a crucial step in the formation of the beta-lactam ring.
So it's like boosting the efficiency of the penicillin assembly line within those fungal cells.
Precisely. Then there's regulation engineering. This involves manipulating regulatory genes to optimize gene expression and overcome bottlenecks. For example, we can modify promoters, the genetic switches that control gene expression, to ramp up the production of those key penicillin-producing genes.
So it's like fine-tuning the genetic control panel to maximize penicillin output. Pretty amazing what we can do with genetic engineering these days.
It is. And we don't stop there. Transport engineering focuses on improving penicillin export from those fungal cells.
Why is that important?
Well, if penicillin builds up inside the cells, it can actually inhibit its own production. It's a feedback mechanism. Ah. So it's like the cells are saying, hey, we've got enough penicillin here, let's slow down.
Exactly. So by improving export, we prevent that buildup and keep those penicillin factories running smoothly.
It's like streamlining the shipping process, making sure those penicillin packages get delivered efficiently.
Precisely. And finally, we have morphology engineering.
Morphology engineering, what's that all about?
This one aims to control the shape and form of the fungal cells, helping to create less viscous broths.
Less viscous, so thinner and easier to mix.
Exactly. This improves oxygen transfer and mixing in those large bioreactors, which is crucial for efficient penicillin production.
It's like optimizing the physical layout of the factory floor to ensure smooth operations.
A great analogy. All these modifications have resulted in modern Penicillium chrysogenum strains that are incredibly efficient. They have high penicillin titers, grow quickly, and are well-suited for industrial-scale fermentation.
Wow, they really are penicillin-producing powerhouses. But even with these optimized strains, the fermentation process itself is still crucial, right? Absolutely. We need to provide the ideal environment for these fungi to thrive and produce those life-saving antibiotics. And that's where the fermentation strategy comes in. For penicillin production, submerged fermentation using the fed batch mode is the gold standard.
Okay, break that down for us. What exactly does that mean?
Submerged fermentation means the microorganisms are grown in a liquid medium within large bioreactors. Think of those giant stainless steel tanks we talked about earlier.
Right, those microbial swimming pools.
Exactly. And fed batch mode refers to how we feed those microbes.
So it's not just a one-time feast at the beginning?
Not at all. In fed batch fermentation, nutrients are added continuously or intermittently throughout the process.
So it's like a carefully controlled drip feed of nutrients to keep those penicillin factories humming. Why is this fed batch mode preferred for penicillin production?
It offers several advantages. First, it helps avoid catabolite repression. Remember how we talked about high glucose concentrations suppressing penicillin biosynthesis?
Right, like glucose was hogging the spotlight and penicillin couldn't shine.
Exactly. Fed batch allows us to carefully control glucose levels, preventing those high concentrations that can hinder penicillin production.
So it's back to that slow and steady wins the race approach.
Precisely. Another advantage is better control over precursor feeding. Phenylacetic acid and its derivatives are essential for penicillin production, but they can be toxic at high concentrations.
So it's a balancing act. We need to provide enough precursor, but not too much.
Exactly. Fed batch allows us to carefully control the feeding of these precursors, optimizing their availability for penicillin synthesis without harming the fungi.
It's like a carefully calculated recipe, adding those ingredients in just the right amounts at just the right time.
A great analogy. And overall, fed batch fermentation often leads to higher penicillin titers compared to other methods.
So it's a more efficient and productive approach. Now let's talk about what those penicillin-producing fungi are actually feasting on in this fed batch setup. What's in that fermentation media?
The media is carefully designed to provide all the essential nutrients for fungal growth and penicillin production.
Okay, let's break it down. What's the primary energy source? What's the carbon source?
Lactose is a common choice nowadays. It provides a slower, more controlled release of glucose compared to using glucose directly.
Ah, so it's back to avoiding those high glucose concentrations that can suppress penicillin production.
Exactly. And we also add phenylacetic acid or its derivatives. Remember, it's a direct precursor for penicillin.
Right. It gets incorporated into the penicillin molecule itself.
Precisely. So it's not just about providing energy, it's also about supplying those building blocks for penicillin synthesis.
Smart. What about nitrogen? What do they use for that?
Corn steep liquor, often called CSL, is a popular choice. It's a cost-effective, complex nitrogen source.
We've heard about corn steep liquor before. It seems like a popular ingredient in these microbial growth media.
It is. It's a byproduct of corn processing, and it's packed with amino acids, vitamins, and minerals. We might also add ammonium salts to ensure a balanced nitrogen supply.
So it's like a nutrient-rich feast for those penicillin-producing fungi. What else is in there?
Well, we need to include other essential nutrients like phosphates and trace minerals, things like iron, manganese, potassium.
Those are important for enzyme function, right?
Exactly. And we might even add antifoams to prevent excessive foaming, which can be a problem in penicillin fermentation.
It sounds like a complex recipe with a lot of different ingredients.
It is. It's all about creating the perfect balance to optimize both fungal growth and penicillin production.
Now, even with the best strains and that optimal fed batch mode, controlling the environment within the bioreactor is still crucial, right?
Absolutely. Things like temperature, pH, and dissolved oxygen levels all need to be carefully monitored and controlled.
Okay, let's break down those critical environmental parameters. First up, temperature. What's the ideal temperature for Penicillium chrysogenum?
Penicillium chrysogenum is a mesophilic fungus, meaning it thrives at moderate temperatures. We typically maintain a temperature range of 25-27 degrees Celsius for optimal growth and penicillin production.
So it's like creating a cozy, temperature-controlled room for those penicillin producers.
Mhm.
But how do we maintain that precise temperature in those massive bioreactors?
Industrial bioreactors have sophisticated temperature control systems. They might have cooling jackets or coils that circulate cool water around the vessel, removing excess heat and keeping the temperature stable.
It's like a giant air conditioner for microbes. Okay, what about pH? What's the ideal pH for penicillin production?
The optimal pH is slightly acidic to neutral, around 6.0 to 6.5. Interestingly, the pH often starts a bit lower, around 5.5, and gradually drifts upward as the fermentation progresses.
So it's a gradual shift in pH over time. Why is that?
Well, that initial lower pH helps prevent contamination.
Ah, so it's back to creating an unfavorable environment for those unwanted microbes.
Exactly. And then, as the fermentation progresses, the pH naturally rises due to the consumption of sugars and the production of organic acids by the fungus.
So it's a self-regulating system. The fungus is essentially adjusting the pH as it grows and produces penicillin. But how do we ensure it stays within that optimal range?
We use automated pH control systems. pH probes constantly monitor the broth, and acids or bases are automatically added to adjust the pH if needed.
It's like a self-driving pH car, constantly making adjustments to stay on course.
Exactly. Now, let's talk about oxygen. We know fungi need oxygen to breathe, just like us. So how do we make sure those penicillin-producing fungi are getting enough oxygen in those large bioreactors?
Yeah, especially with all those fungal cells growing and multiplying.
Right. Penicillium chrysogenum needs a constant oxygen supply. If oxygen levels drop too low, penicillin production will suffer.
So it's all about keeping those penicillin-producing engines fueled with oxygen. How do we achieve that?
Industrial bioreactors use a combination of aeration and agitation.
We've heard those terms before.
Yes, aeration involves pumping air, or sometimes pure oxygen, into the broth. And agitation involves mixing the broth using impellers, those rotating blades inside the bioreactor.
So it's like creating a whirlpool of oxygen-rich broth, making sure those fungi are surrounded by oxygen.
Exactly. But there's a balance to be struck. Too much agitation can actually damage those delicate fungal cells.
So we need to optimize aeration and agitation to maximize oxygen transfer without harming those penicillin producers.
Precisely. It's a delicate balancing act.
Now, with all this talk about controlling the bioreactor environment and optimizing nutrient delivery, I'm curious, how does penicillin actually get made within those fungal cells? What's the step-by-step process?
That's a great question. Penicillin biosynthesis is a multi-step pathway. a series of enzymatic reactions within those penicillium chrysogenum cells. And it all starts with precursor synthesis, the production of three key amino acids, L-alpha aminoadipic acid, L-cysteine, and L-valine.
Amino acids, those are the building blocks of proteins.
Exactly. And in this case, they're the building blocks of penicillin.
So it's like gathering the raw materials for penicillin construction.
Yeah.
Where did these amino acids come from?
Penicillium chrysogenum synthesizes these amino acids from simpler precursor molecules through a series of metabolic reactions. It's like those fungal cells are running their own amino acid factories.
Amazing.
Yeah.
So we've got our amino acid building blocks. What happens next? How are they assembled into that iconic penicillin structure?
The next step involves linking those three amino acids together to form a tripeptide.
A tripeptide.
It's basically a chain of three amino acids, and this particular tripeptide is called ACV for short.
ACV. Okay, so that's like the foundation of the penicillin molecule.
Exactly. And the enzyme that creates ACV is called ACV synthetase.
ACV synthetase. So it's the construction worker that puts those amino acid building blocks together.
Precisely. Now we've got our ACV tripeptide, the foundation for penicillin. The next step is where things get really interesting. We need to form that characteristic beta-lactam ring, which is what gives penicillin its antibacterial activity.
The beta-lactam ring, right? It's like the defining feature of penicillin.
Exactly. And the enzyme responsible for this crucial step is called isopenicillin N synthase, or IPNS for short.
IPNS, the beta-lactam ring builder.
Precisely. IPNS takes that ACV tripeptide and transforms it into isopenicillin N, which contains that essential beta-lactam ring.
Okay, so we've got our isopenicillin N core. Is that it? Is that penicillin?
Not quite. The penicillin story doesn't end there. We need to modify that isopenicillin N molecule to create different types of penicillin.
Different types of penicillin.
Yes, penicillin G, penicillin V, and many others. And this modification is done by enzymes called acyltransferases.
Acyltransferases, what do?
They transfer acyl groups, which are basically chains of carbon and hydrogen atoms, to the isopenicillin N molecule, replacing the original side chain.
So it's like swapping out different accessories on the basic penicillin structure.
Exactly. And the specific acyl group that's added determines the type of penicillin that's produced.
So different acyl groups, different penicillin properties.
Precisely. They might have different spectrums of activity, meaning they're effective against different bacteria or different pharmacological properties, like how they're absorbed and metabolized in the body.
Wow, so by simply swapping out those side chains, we can create a whole family of penicillin antibiotics. It's like a microbial mix and match game.
It is, and that's why understanding the biosynthesis pathway is so important. It helps us understand how these different penicillins are made and why they have different properties.
This has been a fascinating deep dive into penicillin production. But before we move on, I have to ask. We've talked about the main steps of penicillin biosynthesis, but is there anything else going on within those cells that we need to know about?
Well, penicillin biosynthesis is a tightly regulated process, influenced by various factors, both genetic and environmental.
Okay, so it's not just a straightforward linear pathway. There are other things that can influence how much penicillin is produced.
Exactly. And understanding these regulatory mechanisms is crucial for optimizing penicillin production.
So let's dive into those regulatory mechanisms. What are some of the key factors that can influence penicillin production?
One important factor is feedback regulation. Penicillin itself can inhibit certain enzymes involved in its own biosynthesis.
So it's like a self-regulating system. Too much penicillin and production slows down.
Exactly. And we also have carbon catabolite repression, which we discussed earlier. High concentrations of glucose can suppress the expression of genes involved in penicillin biosynthesis.
Right, it's like glucose is saying, hey, focus on me, not penicillin.
Exactly. And to overcome this, we often use lactose as the carbon source in penicillin fermentation.
Lactose, the sugar found in milk.
Right. It's broken down into glucose and galactose, providing a slower release of glucose, which helps prevent that catabolite repression.
So it's back to that slow and steady approach to keep those penicillin factories running smoothly.
Precisely. And besides those metabolic regulatory mechanisms, various environmental factors can also influence penicillin biosynthesis, such as pH, temperature, dissolved oxygen levels, and the availability of those precursors, like phenylacetic acid.
So it's a complex interplay of factors, both internal and external to those fungal cells, that ultimately determines how much penicillin is produced.
Exactly. It's a fascinating and challenging process, requiring careful monitoring and control to ensure optimal penicillin production.
Absolutely. Now, we've talked a lot about strain improvement and optimizing those fermentation conditions, but are there any other strategies scientists use to boost penicillin production?
Definitely. There are a few other tricks up their sleeves. For example, optimizing the feeding strategy for those precursors can make a big difference.
A precursors, those building blocks for penicillin.
Exactly. By carefully controlling the timing and amount of phenylacetic acid added to the broth, we can further enhance penicillin production.
So it's like a finely tuned delivery service, bringing in those penicillin building blocks at just the right time.
Precisely. And some processes even use a two-phase fermentation strategy.
Two-phase fermentation.
This involves growing the fungus in a growth phase, focusing on biomass accumulation, followed by a production phase, where we tweak the conditions to favor penicillin biosynthesis.
Ah, so it's like giving those fungal cells a training regimen, building up their strength before sending them into the penicillin production competition. Are there any other strategies?
Yes, we can even use continuous fermentation, where we constantly feed fresh nutrients and remove product-containing broth, maintaining a steady state of penicillin production.
So it's like a penicillin production assembly line,
Eh,
continuously churning out those life-saving antibiotics.
Exactly. And all these strategies, combined with those optimized strains and controlled conditions, have truly revolutionized penicillin production.
It's a remarkable testament to the power of biotechnology. Now, before we move on to another essential antibiotic, any final thoughts for our listeners on mastering this topic?
Focus on understanding the interplay between strain improvement, the fermentation mode, media composition, and those crucial environmental parameters. If you can explain how these factors work together to maximize penicillin production, you'll be well prepared for your exam.
Excellent advice. Now, ready to explore another antibiotic superstar?
Absolutely. Let's talk about streptomycin.
Perfect. Let's tackle this question from our list. Give an account of streptomycin antibiotics. Now, I know streptomycin is another powerful antibiotic, but what makes it unique? What's its claim to fame?
Streptomycin is an aminoglycoside antibiotic. This means it belongs to a family of antibiotics that are particularly effective against gram-negative bacteria.
Gram-negative bacteria. I remember learning about those.
Yeah.
They're the ones with that extra outer membrane that makes them tougher to kill.
Exactly. That outer membrane acts as a barrier, making it harder for some antibiotics to get inside and do their job.
So streptomycin has a way of getting past that outer membrane.
It does. It targets a different part of the bacterial cell, the ribosome.
The ribosome. That's where proteins are made, right?
Exactly. And by binding to a specific part of the ribosome, the 30S subunit, streptomycin disrupts protein synthesis. It essentially shuts down the bacterial protein production line.
So it's like throwing a wrench into the bacterial machinery. No proteins, no life.
Precisely. And this mechanism of action is particularly effective against those tricky gram-negative bacteria. That's why streptomycin is so valuable for treating infections like tuberculosis, plague, and tularemia.
Wow, it sounds like streptomycin has saved countless lives. Now, let's talk about how it's produced. What microbe is responsible for this life-saving antibiotic?
Streptomycin is produced by Streptomyces griseus.
Streptomyces griseus. Is that another fungus?
It's actually a bacterium, but it's a filamentous bacterium, meaning it grows in those long branching filaments, kind of like a fungus.
Interesting. So we've got a filamentous bacterium producing this powerful antibiotic.
Right. And it belongs to a group of bacteria called actinobacteria, which are known for producing a wide variety of antibiotics and other useful compounds.
So it's like they're the master chemists of the microbial world. Now, I'm guessing we're not just relying on wild-type Streptomyces griseus strains to produce streptomycin in those factories.
Yeah.
Have scientists been working on strain improvement for this antibiotic as well?
Absolutely. Just like with penicillin, strain improvement programs have played a key role in boosting streptomycin production.
So it's back to those microbial breeding programs, selecting for those high-yielding strains. What were some of the techniques used to improve streptomycin production in Streptomyces griseus?
Both classical and modern techniques have been employed. Classical mutagenesis, using UV radiation or chemical mutagens, was used extensively in the early days.
So they were inducing those random mutations and then screening for those rare high-performing mutants.
Exactly. They were looking for mutants that produced more streptomycin, grew faster, or had better morphology for industrial fermentation.
And with the advent of genetic engineering, scientists now have more precise tools for manipulating those streptomycin-producing pathways, right?
Exactly. Genetic engineering has opened up a whole new world of possibilities for strain improvement. We can now target specific genes and pathways involved in streptomycin biosynthesis.
So it's like we've gone from using a shotgun to using a laser pointer. What are some examples of those genetic engineering strategies?
One common approach is gene overexpression. By increasing the expression of key genes in the streptomycin pathway, we can ramp up production.
So it's like we're turning up the volume on those streptomycin-producing genes.
Precisely. And another approach is pathway optimization. This involves modifying the entire metabolic pathway that leads to streptomycin production.
So it's like we're fine-tuning that metabolic assembly line, making sure everything is running smoothly and efficiently.
Exactly. We might reduce flux towards competing pathways, increase precursor availability, or optimize regulatory elements. And then we have regulation engineering, where we modify regulatory genes to remove those brakes on streptomycin production.
It's like we're giving those streptomycin-producing pathways the green light. Now, once we have those optimized Streptomyces griseus strains, we need to create the ideal environment for them to thrive and produce streptomycin. What kind of fermentation setup is used for this antibiotic?
Like with penicillin, submerged fermentation is the method of choice. We cultivate Streptomyces griseus in those large bioreactors, carefully controlling the conditions to maximize streptomycin yields.
Back to those microbial swimming pools. And I'm guessing fed batch fermentation is the preferred feeding strategy.
You guessed it. It offers the same advantages as with penicillin. Better control over nutrient levels and prevention of those inhibitory substrate buildups.
So it's all about that slow and steady approach to keep those Streptomyces griseus factories happy and productive. Now let's talk about the specifics of that fermentation media. What are we feeding these streptomycin-producing bacteria?
The media composition is carefully designed to provide all the essential nutrients for growth and streptomycin production. For the carbon source, we often use glucose or molasses.
Those are good sources of sugar, providing that energy boost for the bacteria.
Exactly. For the nitrogen source, we typically use soybean meal or corn steep liquor.
Corn steep liquor again. It really is the multivitamin of microbial growth media.
It is. And of course, we need to include essential minerals and trace elements.
Right, those are important for various cellular processes and enzyme function.
Precisely. And the specific media composition is often optimized through experiments to maximize streptomycin production while minimizing costs.
It's like a culinary adventure, finding that perfect recipe for those streptomycin-producing bacteria. Now, we've talked about the media, but we also need to create the right environment within the bioreactor, right? What are those crucial environmental factors for streptomycin production?
Just like with penicillin, controlling temperature, pH, and dissolved oxygen levels is crucial for maximizing streptomycin production.
Okay, let's start with temperature. What's the sweet spot for Streptomyces griseus?
Streptomyces griseus also thrives at moderate temperatures. We usually maintain a range of 25–30 degrees Celsius for optimal growth and streptomycin production.
Okay, so similar to Penicillium chrysogenum.
Right. And we use those sophisticated temperature control systems in those bioreactors to maintain that precise temperature range.
Those microbial climate control systems. What about pH? What does Streptomyces griseus prefer?
Streptomyces griseus likes things a bit more alkaline. The optimal pH range for streptomycin production is around 7.0 to 7.5.
So a slightly alkaline environment, and we use those automated pH control systems to keep it stable, right?
Exactly. We want to make sure those enzymes involved in streptomycin biosynthesis are happy and functioning efficiently.
Now let's talk about oxygen. We know Streptomyces griseus is an aerobic bacterium, so it needs oxygen to breathe and grow. How do we ensure those streptomycin-producing bacteria are getting enough oxygen in those bioreactors?
Maintaining high dissolved oxygen levels is crucial for streptomycin production. It's required for both bacterial growth and the synthesis of those streptomycin molecules.
So we need to make sure those streptomycin factories are well ventilated.
Exactly. And we achieve that through aeration and agitation, just like with penicillin fermentation. We sparge air, or sometimes pure oxygen, into the bioreactor. And those impellers keep the broth well mixed, enhancing oxygen transfer and creating a homogeneous environment.
It's all about creating that oxygen-rich whirlpool. But too much agitation can be harmful, right?
Yes, too much agitation can create shear stress, which can damage those Streptomyces griseus cells. So we need to strike that balance, optimizing aeration and agitation to provide enough oxygen without stressing those bacterial cells.
It's all about finding that sweet spot. Now, how long does this streptomycin production process typically take?
Streptomycin fermentation is a bit slower than penicillin fermentation. It usually takes about 5 to 10 days, sometimes even longer, depending on the strain and the specific conditions.
So it's a bit more of a slow burn compared to penicillin.
Yes, it's a characteristic of secondary metabolite production. Streptomycin, like many other antibiotics, is a secondary metabolite, meaning it's not essential for the bacterium's growth, but is often produced in response to specific environmental cues or stresses.
So it's like Streptomyces griseus is taking its time, building up its defenses, and then releasing those streptomycin weapons.
Precisely. And during this extended fermentation period, those Streptomyces griseus cells are busy synthesizing those complex streptomycin molecules through a multi-step biosynthetic pathway, just like with penicillin.
So it's a complex dance of biochemical reactions,
Yeah.
all carefully orchestrated to produce those life-saving molecules.
Exactly. And all those efforts to optimize the strain, the media, and the fermentation conditions pay off in the end, resulting in high yields of this essential antibiotic.
It's truly remarkable how we've harnessed the power of these microbes to produce such valuable medicines. Now, before we move on to another fascinating biomolecule, any final tips for our listeners on acing those exam questions about streptomycin production?
I would say focus on the unique characteristics of Streptomyces griseus and how those characteristics influence the fermentation process. Highlight the importance of strain improvement, media composition, and controlling those key environmental parameters.
Excellent advice. Now, ready to explore a lesser-known but equally important group of enzymes?
Absolutely. Let's dive into the world of pectinases.
Perfect. Let's tackle this question from our list. Write a note on pectinase production and its application. Now, I have to admit, I'm not as familiar with pectinases. Can you give us a quick introduction? What are pectinases and what makes them so special?
Pectinases are a group of enzymes that break down pectin.
Pectin.
Pectin is a complex carbohydrate found in plant cell walls, especially in fruits. Think of it like the glue that holds those plant cells together.
Ah, so pectin is what gives fruits their texture.
Exactly. And pectinases are the enzymes that can break down that pectin.
Okay, so they're like pectin-destroying enzymes. But why do we need to break down pectin? What's the point of that?
Breaking down pectin has various applications, particularly in the food industry. One of the most common uses is in juice clarification.
Juice clarification. How do pectinases help with that?
While that pectin that gives fruits their texture can also make fruit juices cloudy and viscous. Pectinases can break down that pectin, making the juice clearer and less viscous, which is more appealing to consumers.
Ah, so pectinases are like the cleaning crew for fruit juices. They get rid of that pectin cloudiness. What other applications do they have?
They're also used in fruit pulp processing, wine production, textile processing, paper production, even animal feed. They're quite versatile.
Wow, it sounds like pectinases are everywhere. But before we get too carried away with their applications, let's talk about their production. How are these pectin-degrading enzymes produced on an industrial scale?
Microbial fermentation, once again, is the key. And for pectinase production, we rely heavily on our fungal friends.
Fungi, those filamentous powerhouses. Are there any particular fungal species that excel at pectinase production?
Aspergillus niger, our old friend, is a top contender. It produces high yields of pectinases, and its GRA status makes it a safe choice for food applications.
Aspergillus niger, the enzyme superstar. It seems like this fungus can do it all.
It's quite talented. But other fungal species are also used, like Aspergillus awamori and Trichoderma rhizae. And for some applications, we might use bacteria, like Bacillus subtilis.
So we've got a diverse team of microbial pectinase producers. But you mentioned that pectinases are a diverse group of enzymes themselves. What are the different types of pectinases and what do they do?
That's a great question. Pectinases are indeed a diverse bunch, and they're classified based on how they act on the pectin molecule. It's like having a toolbox full of different tools, each designed for a specific pectin-degrading task.
Okay, I'm intrigued. Let's open up that toolbox and see what's inside. What are the main types of pectinases?
One important group is the depolymerizing pectinases. They break down the backbone of the pectin molecule, those long chains of sugar molecules.
So they're like the demolition crew, breaking those pectin chains into smaller pieces.
Exactly. Within this group, we have polygalacturonases, or PGs. They target a specific type of bond in the pectin molecule, the alpha-1 Viro-4 glycosidic linkage.
So they're like specialized bond breakers.
Precisely. And PGs are further classified based on how they attack those bonds. Some start at the end of the chain, others attack randomly from within.
It's amazing how these enzymes have evolved such specific modes of action. What other types of pectinases are there?
Another important group is the pectin liases, or PLs. They also break those alpha-1 variophoric glycosidic linkages, but use a slightly different mechanism.
Okay, so different tools for the same job.
Right. And then we have the pectin esterases, or PEs. They remove methyl groups from the pectin molecule, making it more susceptible to degradation by those depolymerizing pectinases.
So it's like they're prepping the pectin molecule for demolition.
Exactly. And there are other pectinases that target the side chains of the pectin molecule, further breaking down those pectin structures.
Wow, it's a whole pectin-degrading party. Now let's shift gears and talk about how these pectinases are produced. You mentioned microbial fermentation as the primary method, but can you elaborate on that? What are the different types of fermentation processes used for pectinase production?
Well, just like with many other industrial bioprocesses, both submerged fermentation and solid-state fermentation are used for pectinase production.
Okay, we've talked about submerged fermentation before, but remind us about solid-state fermentation. What's the difference?
In submerged fermentation, those microorganisms are grown in a liquid medium, typically in those large bioreactors. In solid-state fermentation, the microorganisms are grown on a solid substrate, like agricultural byproducts that are rich in pectin.
So like those microbes are growing on a bit of pectin-rich material.
Exactly, and each method has its pros and cons.
What are some of those pros and cons?
Submerged fermentation offers better control over the fermentation conditions and is easier to scale up for industrial production.
So it's the more controlled and scalable approach.
Right. Solid-state fermentation, on the other hand, is more cost-effective and environmentally friendly. It utilizes those agricultural wastes that would otherwise be discarded.
So it's a more sustainable approach. But which method is more commonly used for industrial pectinase production?
Submerged fermentation is still the dominant method for large-scale pectinase production.
Why is that? What are the limitations of solid-state fermentation?
Solid-state fermentation can be more challenging to control, and scaling up can be more complex.
Okay, so it's a trade-off. Control and scalability versus cost-effectiveness and sustainability.
Exactly. But regardless of which method we use, choosing the right microorganism is still crucial.
Right, we need those pectinase-producing champions. So what factors do scientists consider when choosing the microbial workforce for this task?
Well, pectinase yield is obviously important. We want a microbe that produces a lot of those pectin-degrading enzymes.
Makes sense. Efficiency is key in industrial production.
Exactly. We also consider enzyme properties. Different pectinases have different optimal pH and temperature ranges.
Right, some enzymes like it hot, some like it cold.
Precisely. And some are more stable than others, meaning they can withstand harsher conditions.
So we need to choose a microbe that produces pectinases with the right properties for the intended application.
Exactly. We also consider substrate utilization, how efficiently a microbe can use those pectin-rich materials.
So we want those microbes to be good eaters, efficiently converting those substrates into valuable pectinases.
Exactly. And increasingly, we also consider genetic amenability.
Genetic amenability, what does that mean?
It means how easily we can manipulate the microbe's genes to further enhance pectinase production or modify enzyme properties.
So we're looking for microbes that are not only good pectinase producers, but also good candidates for genetic optimization. It's like we're scouting for those microbial all-stars with the potential to become even better.
Precisely. Now, once we've chosen our microbial team and the fermentation method, we need to provide those microbes with the right food to thrive and produce those pectinases. That brings us to the fermentation media, the nutrient broth that feeds those pectinase-producing factories.
Okay, let's get cooking. What's in this pectinase-boosting recipe?
Well, like with other enzyme production processes, we need a good balance of nutrients, and the exact recipe depends on the microorganism, the fermentation method, and the specific pectinase we're trying to produce.
So it's a customizable menu for those pectinase-producing microbes. What are some of the common ingredients?
For pectinase production, we often include pectin-rich substrates in the media, things like citrus peel, apple pomace, sugar beet pulp, those agricultural byproducts we talked about.
So it's back to utilizing those waste materials and turning them into something valuable.
Exactly. It's a win-win for sustainability and cost-effectiveness. But besides pectin itself, we might also include other carbon sources, like glucose or glycerol, to support microbial growth.
So it's a balanced diet with pectin as the main course. What about nitrogen?
We often use organic nitrogen sources like yeast extract, peptone, or corn steep liquor.
Corn steep liquor making another appearance.
It's a popular ingredient. It provides those amino acids and other growth factors that those microbes need.
And of course, we can't forget those essential minerals and trace elements.
Exactly. We need those for various cellular processes and enzyme function. It's all about providing the right nutrients in the right amounts to keep those pectinase-producing factories running smoothly.
Now, we've talked about the media composition, but what about the environment within the bioreactor? Do we need to control temperature, pH, and oxygen levels for pectinase production as well?
Absolutely. Those environmental parameters are just as important for pectinase production as they are for antibiotic production.
Okay, so let's start with temperature. What's the ideal temperature range for those pectinase-producing microbes?
Most of them are mesophilic, meaning they prefer moderate temperatures. The optimal range is usually somewhere between 25 and 37 degrees Celsius.
So back to those temperature-controlled environments.
Right. But remember, the exact temperature can vary depending on the specific microbe and the pectinase we're producing.
Okay, so we need to fine-tune the temperature based on our microbial workforce. What about pH?
The optimal pH also depends on the microorganism and the specific pectinase. Some prefer slightly acidic conditions, others prefer near neutral or slightly alkaline conditions.
So it's another parameter that needs to be carefully adjusted.
Exactly. And of course, we can't forget about oxygen. Most pectinase fermentations are aerobic, so those microbes need oxygen to breathe.
Right, so we need to make sure those bioreactors are well aerated.
Exactly. We can use those aeration and agitation techniques we discussed earlier to maintain optimal oxygen levels.
It's all about keeping those pectinase-producing factories well ventilated. Now, how long do these pectinase fermentation processes typically take?
The duration can vary, but in submerged fermentation, it usually takes somewhere between 2 and 7 days.
Okay, so it's a bit quicker than those antibiotic fermentations.
Yes. And solid state fermentation usually takes a bit longer, maybe up to 2 weeks or more.
So it's back to that trade-off between speed and sustainability.
Right. But regardless of the method or the duration, once those microbes have produced those pectinases, we need to extract and purify them.
Right, it's time to harvest those pectinase gems. What are the key steps in this downstream processing adventure?
Well, the first step is usually solid-liquid separation. We need to remove those microbial cells or that solid substrate from the liquid containing those precious pectinases.
So it's back to separating the wheat from the chaff. How do we do that?
Filtration or centrifugation are commonly used. For submerged fermentation, we might use those large rotary vacuum filters or pressure filters. And for solid-state fermentation, we might use a combination of filtration and pressing to extract those pectinases from the solid material.
So it's all about finding the right method to separate those solids from those liquids.
Exactly. And once we have a clarified pectinase solution, we often move on to enzyme concentration.
Enzyme concentration.
We want to reduce the volume of that solution, making it more concentrated.
So it's like we're making those pectinases more potent.
Precisely. This makes the subsequent purification steps more efficient.
What methods are used for enzyme concentration?
Several techniques can be used, like ultrafiltration, precipitation, or evaporation. Each method has its own advantages and limitations.
So it's like choosing the right tool for the job. And once we have that concentrated solution, we can move on to the main event, purification.
Exactly. Purification is all about removing any remaining impurities to get that high-purity pectinase product.
And what's the purification method of choice for pectinases?
Chromatography is often used. We might use ion exchange chromatography, gel filtration chromatography, or affinity chromatography.
Ah, those chromatography techniques are back. Can you quickly remind us how those methods work?
Sure. Ion exchange chromatography separates molecules based on their charge. We use a stationary phase with specific charges, and molecules with opposite charges bind to it while others pass through.
Right, it's like those charged beads are attracting those pectinase molecules with opposite charges.
Exactly. Gel filtration chromatography separates molecules based on size. Smaller molecules can penetrate those pores in the stationary phase and take a longer path, while larger molecules pass through more quickly.
So it's like a size-selective filter.
Right. And affinity chromatography utilizes specific binding interactions. We use a ligand, a molecule that binds specifically to our target pectinase, immobilized on a stationary phase, Only those pectinases that bind to that ligand will be captured and purified.
So it's a highly specific purification method.
Exactly. And sometimes we might even use a technique called aqueous two-phase extraction.
Aqueous two-phase extraction, what's that?
It uses two immiscible water-based phases to separate biomolecules. It's a gentle technique that can be very effective for pectinase purification.
Okay, so we've got a whole arsenal of purification techniques at our disposal. So after all these purification steps, we finally have our pure pectinase product, right? Are we done yet?
Almost. The final step is usually enzyme formulation and stabilization.
So it's time for those pectinases to get ready for their big debut.
Exactly. We want to create a stable and user-friendly product, whether it's a liquid or a solid formulation.
Okay, so what are some common formulation and stabilization strategies for pectinases?
For liquid formulations, we typically dissolve those purified pectinases in a buffer solution with added stabilizers to prevent them from clumping together and losing activity.
So it's like giving those pectinases a protective bubble bath.
Right. And for solid formulations, we often dry those pectinases, creating a powder that's more stable and easier to handle.
So it's like transforming those pectinases into a convenient and shelf-stable form.
Exactly. And there are different drying methods, like spray drying and freeze drying.
So it's like choosing the right packaging method to preserve those precious pectinases. Now we've covered pectinase production from start to finish, from those raw materials to those purified enzymes. Let's shift gears and explore their amazing applications.
Sounds good.
We've talked about their role in juice clarification, but you mentioned they're used in many other industries as well.
Right. Pectinases are incredibly versatile. In the food industry, they're used in fruit pulp processing, wine production, and even vegetable processing. They can help soften vegetables, improve texture, and make peeling easier.
So they're like tiny culinary assistants helping us prepare our fruits and vegetables.
Exactly. And pectinases aren't limited to the food industry. They're also used in the textile industry for bioscouring, a process that removes impurities from natural fibers like cotton and flax.
So they're like a natural cleaning crew for fabrics.
Precisely. And they're used in paper production, helping to break down those plant cell walls in wood pulp and even de-inking recycled paper fibers. They're even used in animal feed to improve the digestibility of plant-based feeds.
Wow, the applications are endless. It seems like these pectinases can do it all.
They're quite impressive. And as research continues, we're likely to discover even more applications for these versatile enzymes.
Now, our final question for today focuses on the downstream processing of protease enzymes,
Okay.
another important class of enzymes with diverse industrial applications.
Yes, they are.
What can you tell us about downstream processing of protease enzymes?
So proteases, as their name suggests, are enzymes that break down proteins into smaller peptides or amino acids. They're essential for a wide range of biological processes, and we've harnessed their power for various industrial applications, from laundry detergents to food processing and pharmaceuticals.
Right. It's all about transforming that crude enzyme mixture from fermentation
Yes,
into a valuable and marketable product.
that's right.
What are the main objectives of protease downstream processing, or DSP?
The primary objective of protease DSP is to recover, purify, and formulate these enzymes into stable and usable forms.
Okay.
This involves a series of steps designed to isolate the target protease from the fermentation broth, remove impurities, concentrate the enzyme solution, and ultimately convert it into a marketable product, whether it's a liquid, powder, or even an immobilized enzyme.
It sounds like a carefully choreographed sequence of operations.
It is.
Can you walk us through the typical steps involved in protease DSP?
Certainly. The first step is usually cell harvesting or biomass removal, using techniques like centrifugation, filtration, or a combination of flocculation and coagulation, which we discussed earlier. If the protease is intracellular, meaning it's produced inside the microbial cells,
Right.
the next step is enzyme recovery or extraction, using various cell disruption methods to release the enzyme.
So those mechanical or non-mechanical lysis methods we discussed earlier might come into play here.
Exactly. Once we've released the protease, we need to concentrate the enzyme solution to reduce its volume and make subsequent purification steps more efficient. This can be achieved using techniques like ultrafiltration, evaporation, or precipitation.
It's like squeezing out the excess water to get a more potent enzyme solution.
A good analogy. Then comes the purification step, where we aim to remove impurities like other proteins, cell debris, pigments, and salts to achieve the desired level of purity for the specific application.
And I imagine the purity requirements vary depending on the intended use.
Absolutely. For some applications, like certain types of laundry detergents, a relatively crude protease preparation might be sufficient.
Okay.
But for more sensitive applications like food processing or pharmaceuticals, a much higher level of purity is essential.
Makes sense.
We can use a variety of chromatography techniques, such as ion exchange chromatography, size exclusion chromatography, affinity chromatography, or a combination of these methods to achieve the desired level of purity.
Chromatography seems to be the workhorse of enzyme purification.
It's an incredibly powerful and versatile tool for separating and purifying biomolecules based on their specific properties.
Hmm.
Once we have our purified protease, the final step is formulation and stabilization, where we convert the enzyme into a stable and user-friendly product.
So this is where we decide whether it'll be a liquid, powder, or something else.
Exactly. We choose the most suitable formulation based on the intended application and the desired shelf life. Liquid formulations are often preferred for applications where ease of handling and dosing is important, while powder formulations tend to be more stable and have a longer shelf life.
Right.
We can also immobilize enzymes, which involves attaching them to a solid support, such as beads, membranes, or fibers.
Immobilized enzymes. What's the advantage of that?
Immobilization offers several benefits. It can enhance enzyme stability, allowing them to withstand harsher conditions, such as high temperatures or extreme pH.
Okay.
It can also make enzymes reusable, as they can be easily recovered from the reaction mixture and used again.
Right.
And it can facilitate continuous processing, where enzymes are packed into a column or reactor, and the substrate is continuously flowed through, enabling a more efficient and scalable process.
It sounds like immobilization is a clever way to enhance enzyme performance and versatility. Now, I imagine that throughout this entire downstream process, quality control is paramount.
Absolutely. We need to constantly monitor enzyme activity, protein concentration, and purity at each stage of the process to ensure we're producing a high-quality product that meets the required specifications. We employ a variety of analytical techniques, including enzyme activity assays, protein concentration assays, SDS page, HPLC, and mass spectrometry to assess the quality and purity of our protease preparations.
It's like having a team of inspectors ensuring that only the best protease makes it to the final product. Well, it looks like we've reached the end of our industrial biotechnology deep dive. We've covered a lot of ground today, from the production of everyday ingredients like citric acid to the complex processes involved in creating life-saving antibiotics and versatile enzymes.
It's been a fascinating journey exploring the world of microbial factories and biochemical transformations. I hope our listeners feel well equipped to tackle those BSc Biotechnology Semester VI exams.
Absolutely. And don't forget, there's always more to explore in the world of industrial biotechnology. It's a dynamic and rapidly evolving field with endless possibilities for innovation and discovery. So stay curious, keep learning, and who knows, maybe one day you'll be designing the next generation of bio-based products that will shape our future.